What's up, Nerdlings? Queen Nerdling here, and today we're going to be talking about one of my favorite topics, nucleic acids. Let's get started. We're going to be talking about DNA, RNA, and soon you'll understand what A, T, U, C, and G are. All right, so nucleic acids encompass DNA and RNA. So the monomer, meaning single unit or one, of nucleic acids is a nucleotide. The polymers are both DNA and RNA. And the type of bond that holds them together is a phosphodiester bond. Now in DNA, we also have a very important bond that holds those nitrogenous bases together, and that's hydrogen bonding. And if you recall from some of my previous lectures, hydrogen bonds are easy to make and easy to break. So as far as the structure of nucleic acids, they're composed of nucleotides. So we have a nitrogenous base that's carbon-nitrogen ring. So if you look right here, we have these carbon-nitrogen rings. And that's what's going to compose that nitrogenous base. Now we also have a 5-carbon or a pentose sugar. So count them 5. 1, 2, 3, 4, and 5 carbons for that pentose sugar. We're also going to have a phosphate group. So phosphorus surrounded by 4 oxygens. And that's what makes up that sugar phosphate backbone of both DNA as well as RNA. So we have 2 different types of nucleotides. We have purines and pyrimidines. Now, the way that I always kind of remember those in my head is that purines sounds like rings, and they have 2 rings. So purines have 2 rings, and those are adenine and guanine. Those are both classified as purines. Pyrimidines only have 1, kind of like a pyramid, all right? So pyrimidines are thymine and cytosine, and they only have 1 ring, while our purines have 2 rings. So as far as their function, obviously they're there to store and transmit genetic information. They're basically the functioning for blueprints and building proteins within our bodies. So they go from DNA, which is typically in the nucleus in eukaryotic cells, and that DNA goes into messenger RNA or gets transcribed into messenger RNA. That messenger RNA is allowed to leave the nucleus where it finds a ribosome, and once at the ribosome, it can work to construct a fully functional protein. Now, this transfers the information from one cell to the next, and that's called our genetic material. So passing along of those genes from one generation to the next. It's a blueprint for new cells, and again, a blueprint for that next generation of cells. So again, our 2 examples are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Keeping in mind that there are slight differences between those 2 nucleotide or the 2 nucleic acids. In DNA, we have a double helix, and in RNA, we have a single helix. In DNA, we do have those hydrogen bonds that are holding those nitrogenous bases or the nucleotide bases together, whereas in RNA, we don't have those hydrogen bonds because it doesn't have a complementary strand. So taking a little bit more of a detailed look at the 2 different types of nucleic acids, over here we have DNA, and as you can see, we have that double helix. On both DNA and in RNA, we have phosphodiester bonds that are linking those sugar phosphate backbones to one another. However, in DNA, between those nucleotide bases, we have those hydrogen bonds. And again, hydrogen bonds are easy to make and easy to break. In DNA, we have adenine, guanine, cytosine, and thymine. And in RNA, we have adenine, guanine, cytosine, and uracil instead of thymine. So looking at DNA and RNA, again, those are polymers, meaning many. So they're composed of many nucleotide bases put together. And they exist in triphosphates. So fun fact, just like we have ATP, which is our universal energy molecule within our body, each of those nucleotide bases brings with it its own source of energy. So when we are forming DNA strands or RNA strands, energy has to be involved to kind of link those together. And so we actually have GTP, guanine triphosphate, CTP, cytosine triphosphate, TTP, thymine triphosphate, UTP, uracil triphosphate, and of course we have ATP, so adenosine triphosphate. And if you look at this, the way that it's broken down, we would have a triphosphate, so 1, 2, and 3 phosphates. We would call this a nucleoside triphosphate. We break off the last one, that's breaking a high energy bond. Whew, we get to use that energy. We'll talk more about that in a different video. And we are left with a nucleoside diphosphate, meaning it has 2 phosphate groups left. We go through that again. We cleave off that second phosphate, releases some energy, not quite as much as that third phosphate, but we still got a little bit left. Then we are left with a nucleoside monophosphate. Now this is going to be that building block for the sugar phosphate backbone / nitrogenous base for our DNA and RNA strands. And if we cleave that phosphate off, we are left with just a nucleoside, just that ribose and the nucleotide base. All right, so just like with so many other different types of structures and different types of functions and different types of organisms, we have similarities and we have differences that fall between DNA and RNA. So some of the similarities between these two nucleic acids are the fact that they both have 5 carbon sugars. They both have phosphate groups, and they both have nitrogenous bases. They are both connected by phosphodiester bonds for their backbone, so their sugar phosphate backbone, and those are covalent bonds. They have 5' and 3' ends. Now when I say 5' and 3' ends, I'm just talking about the position of the carbon on those 5 carbon sugars. So if we go around in a ring, we can count 1, 2, 3, 4, and 5. And depending upon the orientation of that sugar, we're either going to refer to it as 3' to 5' or 5' to 3'. And again, that just has to deal with the sugars and the carbon placement on those sugars in the sugar phosphate backbone. So both of them have nitrogenous bases that are perpendicular to the backbone, to that sugar phosphate backbone. Now, obviously, there are many differences between DNA and RNA. So DNA is deoxyribose sugar, RNA is just ribose sugar. DNA has thymine, while RNA has uracil that pairs with adenine. DNA is double-stranded, whereas RNA is single-stranded. And because of this, DNA's structure is more stable than RNA. DNA in eukaryotic cells remains in the nucleus. It will not leave. Whereas in eukaryotic cells, RNA can actually leave the nucleus. DNA's double strands run anti-parallel. So we have different types of RNA. We're going to talk a lot more about this when we get into protein synthesis, but just kind of a little super quick synopsis. We have messenger RNA, which is located inside the nucleus. It forms a complementary strand to the DNA because it needs to take the message out of the nucleus. Once the messenger RNA has that information from the DNA, it exits the nucleus. It finds a ribosome. Now, ribosomes are composed of rRNA, or ribosomal RNA. So messenger RNA will come over here to the ribosome, and then we have tRNA, our transfer RNA. Transfer RNA carries the anticodon, which will match up with 3 nucleotide bases on the messenger RNA strand. Now, those 3 nucleotide bases on the messenger RNA strand are called codons. So the anticodon will bind with the codon through hydrogen bonding. It also carries with it an amino acid. So each of those transfer RNAs will come to the ribosome. The messenger RNA strand will be in the ribosome, and those transfer RNAs are going to drop off their amino acids. When that's occurring, there's going to be peptide bonds that form between each of those amino acids, forming a fully functional protein. So the whole process of protein synthesis is oftentimes referred to as the central dogma. So that's taking into consideration that we're going from DNA in the nucleus, and somehow we have to get the message from the DNA out of the nucleus so it can make the blueprints for a cell, or so it can make those blueprints to form an enzyme that we need to use to break down some type of biological molecule that we've consumed and need to utilize within our body. So the process by which DNA is transcribed into messenger RNA is called transcription. Now, once that occurs, messenger RNA can leave the nucleus, and then translation will occur. It will latch on to a ribosome, and it will be translated into a fully functional protein. So the protein assembly goes to the ribosome. We have ribosomal RNA. Messenger RNA is taking the message from the nucleus, from the DNA in the nucleus outside, and then transfer RNA is bringing those anticodons and the amino acids, and they put them together, and it makes the primary sequence of a protein, which is just that sequence of amino acids. The protein then assembles into secondary, tertiary, and sometimes quaternary structure based on those R group interactions after it's released from the ribosome. So I'm going to do a super quick recap when we're talking about the structures of a protein. So when I'm talking about the central dogma and forming a protein, we have several different structures of a protein. So at the ribosome, we're forming amino acids. Those transfer RNA molecules are bringing with it an amino acid, so one each. As those transfer RNA molecules are bringing their amino acids to the ribosome, those amino acids start to link together through peptide bonds. So this would be one amino acid, 2, 3, 4, so on and so forth, until we get this long chain of amino acids. Since there are many proteins, or many amino acids in this protein, I should say, we call it a poly, meaning many, peptide chain. And each of those amino acids is linked through a peptide bond. Now, all of those amino acids linked together is the primary structure of a protein. Now, when we get into secondary structures, we have two. We have an alpha helices, very similar to the structure of DNA and RNA. So we get this alpha helices, and the little rings are connected through hydrogen bonds. This is one type of secondary structure. The other type is called a beta-pleated sheet. And it's basically kind of this up-down structure, again, held together through hydrogen bonds. Now, we have a third structure, or a third level of structure of a protein. That's called the tertiary structure. And we start to get bonds, and it starts to look like a squiggly. So we're forming stronger ionic or salt bonds, and we're also forming what's called disulfide bridges. And those are very strong covalent bonds that help maintain the shape of the protein. Sometimes we have two or more of these tertiary structures that come together, and we call that a quaternary structure, and that's also a fully functional protein. Now, if I add extreme heat to this, it denatures and goes back into its primary conformation. So that was your quick recap of the structures. So now that we've kind of had a little review about the different conformations or different structural levers of a protein, we're going to move on to that central dogma. So very quickly, we have transcription. Transcription occurs inside the nucleus. So we are transcribing the message from DNA into messenger RNA. So we have our DNA up here. We're going to have a messenger RNA template that forms. I'm not going to go through exons and introns now, but just know that exons get spliced together for the final messenger RNA strand, and exons get to exit the nucleus because we want to keep some of the bad stuff inside. We don't always need everything that that DNA strand codes for. So this is the messenger RNA strand, exits the nucleus, also needs a G cap, meaning guanine, and a poly A tail, meaning lots of adenosine molecules. So put a cap on it, slap a tail on it, poly A tail, and now it gets to leave the nucleus. So this then travels to a ribosome. once this messenger RNA strand is at the ribosome, now translation into a protein can occur. We have transfer RNA molecules that are going to take the codon, or the anticodon, I should say, and that's going to link up to the codon on that messenger RNA strand. So transfer RNA has an amino acid on top. It has an anticodon on the bottom. That anticodon links through hydrogen bonds to the codon on the messenger RNA strand. As this guy comes in, the transfer RNA, it forms those hydrogen bonds. It will drop its amino acid off, and that amino acid will become linked to the amino acid next to it through a peptide bond. Now, those peptide bonds are much stronger than the hydrogen bonds because hydrogen bonds aren't easy to make and easy to break. So those hydrogen bonds are broken, transfer RNA leaves to pick up another amino acid, and it leaves its initial amino acid as part of that growing polypeptide chain that will eventually become a fully functional protein. So sometimes mutations occur. Nothing can be perfect 100% of the time. So sometimes we get mutations in the sequence, and these are changes in the nucleotide sequence of a nucleic acid. Sometimes they can be good. Most of the time, they're either bad or they're silent. And these changes can cause a change to the protein structure. Now, if we change the structure of something, most of the time, it's going to change the function. Every once in a while, it makes the function better, but most of the time, it makes the function worse, or it renders it completely unfunctional. So a common example of a mutation is sickle cell anemia. This is a substitution or a point mutation, meaning there's a single nucleotide base that gets substituted at a point for another nucleotide base. So looking at this sequence right here, we're going to be focused on this right here, this triplet, which is called a codon. So in this codon, we have CTG. Now this is the template DNA strand. We have to transcribe that message into messenger RNA. So we transcribe it into messenger RNA and we get GAG. We use a codon chart and we find out that that codes for glutamic acid. We have this long line of nucleic acids that's coding for a functional protein. Now we get a mutation in this. So now in the DNA strand, and instead of it saying CTC, we now have CAC. Now the complementary messenger RNA strand now gets skewed, and now that complementary messenger RNA strand is GTG. Now instead of GTG coding for glutamic acid like GAG does, GTG codes for valine. Now this small little mutation causes a structural change in that protein, and that structural change changes the function of that. Now, it will still form a red blood cell, but that red blood cell is not going to work as well as a normal red blood cell. And because of this mutation, it causes a change in the shape of that red blood cell. So instead of being this nice disc shape, now we get a sickle shape. And that is why they call it sickle cell anemia. So in review for our nucleic acids, we all know now that they are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So the monomer of nucleic acids is a nucleotide base with a 5-carbon sugar and a phosphate. There are 5 different nitrogenous bases. We have adenine, thymine, guanine, and cytosine. Those are all in DNA. Now in RNA, we have adenine, uracil, guanine, and cytosine. The polymer forms are RNA and DNA, and the function of both RNA and DNA are to transmit and house genetic information. And this controls everything within the cell, the building blocks for protein, and it's basically what makes me me and what makes you you. So I hope that was helpful. If you want more quirky little educational videos, you can go to www.nerdlingscience.com. Have a fantastic day. This is the Queen Nerdling signing off.